Hi, in this video it's part 2. I will talk about eukaryotic cell components. First one was prokaryotic cell components. And within eukaryotic cells they have two different types. So one is plant cell and the other one is animal cell. I will talk about the similarities between prokaryotic cells and eukaryotic cells first, and then I will go in detail about these two. I made a video on differences between plant and animal cell. That's another video, so you can look at it if you're curious. Otherwise, we'll talk about cell components here. So both prokaryotic and eukaryotic cell contains cell wall. With the exception, animal cell doesn't carry cell wall, and in plant cells they do. And in plants, cell wall is made out of cellulose, which is a glucose molecule connected differently in long chains, and that's why in plants they are considered polysaccharide. So cellulose is a polysaccharide, whereas in prokaryotic cells it's made out of peptidoglycan, and there are more peptides like proteins added together, like amino acid and the sugar molecules together. So that's the difference. And their function is same. It's a rigid, strong outer layer which protects the cell from osmosis, and it supports the cell by maintaining their shapes. And in cytoplasmic membrane, plasma membrane, cell wall membrane is also present in eukaryotic and prokaryotic cells, and it's a barrier that separates the inside and cell from the outside of the cell. And their function is to control which nutrients can enter or exit. So they can kick out the waste, and they can also bring in some nutrients, but only want to bring in some of the necessary ones. And so they would actually carry some kind of transport proteins on their plasma membranes to allow that process. And all of them have cytoplasm, which is the aqueous mixture containing macromolecules, ions, proteins, ribosomes, and so on, which fills the dead space inside the cell. The molecules found in cytoplasm are essential for efficient metabolism. And all of them contain ribosomes. These are actually the free ribosomes. Prokaryotic cells contain free ribosomes, and these are usually found within the cytoplasm, and it's the site of protein synthesis. And now the difference comes from, so eukaryotic cells also have endoplasmic reticulum where ribosomes are bound to. So that's the difference between prokaryotic and eukaryotic cells. And finally, flagellum. It's actually exception. Most plant cells and animal cells don't contain flagellum, but exception that it's in plants, the protozoa actually have found to be carrying some flagellum, and which is a long microtubule structure that allows cells to move. So that's really most of the similarities have between eukaryotic and prokaryotic cells. Now we'll go in detail about eukaryotic cells. Now components only specific to these cells are a nucleus, so you can see this lovely circle up here and here. It's a nucleus, which is the largest organelle in eukaryotic cells. These contain DNA, and it's the site of mRNA synthesis. And it's a two-nuclear membrane, both made out of phospholipid bilayer. And inner membrane and outer membrane are connected to their rough endoplasmic reticulum. The dense DNA region is the nucleoplasm, and it can have fibrous proteins, which are lamins, which are the ones giving nucleus its shape, and it allows DNA to be bound inside nucleus. And both of them contain nucleolus, which is a sub-organelle of the nucleus found inside nucleus, like here. It's the site of synthesis for ribosomal RNA and ribosomal subunit for protein synthesis that will be used later. Both cells contain mitochondria, which can occupy up to 25% of cytoplasmic space. It's a small organelle which is a site of electron transport chain for ATP production during aerobic metabolism, and also inside mitochondria in the matrix, tricarboxylic acid cycle occurs, which is a step that's preparing necessary precursor molecules for biosynthesis and also to prepare for electron transport chain. And they contain intermembrane space between outer membrane and inner membrane, and it's very similar to gram-negative bacteria. So the scientists and researchers suggest that a theory called endosymbiosis, where probably a big bacteria or their cell engulfed small bacteria, so that's why they are inside the cells here. And this is because DNA are found within mitochondria. It's their own DNA, which they would often use to repair themselves, because mitochondria contains a lot of oxidative species that could damage their own structure much. Chloroplast is only found in plant cell. That's the difference between plant and animal cell. Chloroplast is similar to mitochondria. They have two membranes, inner membrane and outer membrane. And inside of chloroplast is filled with thylakoid sacs, often held in matrix called stroma in grana stacks. The green pigments, the chlorophylls and thylakoid membranes and other pigments and enzymes are used for ATP synthesis during photosynthesis, and plants use light as their energy source. Chloroplast also contains their own DNA, and it's similar to cyanobacteria. That's what their research have suggested. Now here's rough endoplasmic reticulum, where it's rough because they have ribosomes bound to them. It's closed interconnected membrane vesicles, and this is rough area, and it's a site of synthesis for membrane organelle proteins and protein to be exported that are to be secreted. And smooth endoplasmic reticulum, it's actually another kind of layer. I just didn't draw another one. You can just kind of see that there is a difference. There's no ribosomes bound here. That's smooth endoplasmic reticulum, and it's the site of fatty acid and phospholipid synthesis. And in liver, enzymes in smooth endoplasmic reticulum can modify or detoxify hydrophobic chemicals like carcinogens, pesticides, by making them more water soluble and into something that is familiar in our body. So these are important. Have important functions in our cells. And here's my lovely Golgi apparatus, or Golgi body. And they process protein that's synthesized in a rough endoplasmic reticulum. And they contain three parts, which is cis, medial, and trans parts. And transfer vesicle from rough endoplasmic reticulum first binds to the cis region, so here, and then they fuse. Then the proteins move to the medial and to trans region. And in these regions, protein is being modified according to their needs because they will be secreted. And some are fused with plasma membrane or secreted outside of the cell. So they can be incorporated inside the plasma membrane, or they can be secreted out for another cell to use them. And vacuoles are here only found in plants. It's site of storage for ions, nutrients, and water. Vacuoles often fill a lot of space inside plant cells. These can up to 90%, sometimes even they can fill up to 90% of the plant cells. And some plants also contain granule, which is an extra storage for starch materials. And centrioles are it's a two pair molecules found only in animal cell, and these are used for cell division, because during cell division one and one, they would separate into each end of the pole, and they would start forming microtubules, like spindle fibers, to separate the chromosomes. So centrioles are important for cell division. And that's pretty much it. So if you need more, if you need detailed explanation, I attached my link to my blog, where I also have references, so if you want to refer back to the original materials that I used to make the post. I hope this helps, and have fun studying.